Welcome. Today we are going to learn about the isolation of the plasmid DNA. The plasmid DNA, which is also called as the extrachromosomal genetic material. This plasmid DNA, it is present into the bacterial cell. Okay, so in the bacterial cell, there are the two types of the DNA they are present. First one, it is the plasmid DNA, and the second one, it is the genomic DNA. So here, when we have to isolate the plasmid DNA, we have to separate out it from the genomic DNA because the plasmid DNA, it is a DNA only, and the genomic DNA, it is also a DNA. That is, both of them, they are the deoxyribose nucleic acid. Okay, so their physical chemical properties, they are the same one. Okay, so here we have to separate out the plasmids from the genomic DNA. So we first have to find out what is the difference between the plasmid DNA and the genomic DNA so that we can separate out the plasmids from the genomic DNA. So here the major difference, it is the size. Size of the plasmid DNA, it is 8% that of the genomic DNA. So the plasmid DNA, it can be separated out, it can be isolated according to its size. And second difference, it is the conformation. The plasmid DNA, it has a different conformation than the genomic DNA. Okay, now here, as we know the differences between the plasmid DNA and the genomic DNA, there are the two methods, okay, for the isolation of the plasmid. The separation on the basis of the size and the separation of the plasmid on the basis of the conformation. So, though these are the two methods which are present for the isolation of the plasmids, the steps in the isolation of the plasmid DNA, they are the similar one. First, we have to grow and harvest the bacterial cells. Then the cells, it has to be broken to release the content. Now, the DNA, where it is present inside the bacterial cell. So first of all, we have to break down the bacterial cell so that there is a release of the DNA into the medium or into the solution, and that cell extract that we have got, from that cell extract we can extract out our DNA. Now, when we say it is a cell, in that cell there is a presence of, okay, in that cell there is a presence of the carbohydrates, lipids, proteins, nucleic acid. Nucleic acids, two types. First one, it is the DNA. Second, it is the RNA. So what we have to isolate first, we have to first isolate the DNA. That is, we have to remove the impurities like the carbohydrates, lipids, and the proteins, and the RNA also we have to remove. So we are getting the DNA. And now here the two types of the DNA, plasmid DNA and the genomic DNA. So we have to extract out, we have to separate out what? Separate out the plasmid DNA from the genomic DNA. So in this way, there are the number of ways by which we are going to separate out the plasmid DNA from the genomic DNA. Now, in the solution, there is a presence of the DNA, plasmid DNA, then we have to concentrate it. And in this way, these are what? These are the steps in the isolation of the DNA. Now, separation on the basis of the size. Now, here you can see it is what? It is a bacterial cell. Okay. Now, when you look at this bacterial cell, okay, this outer one, it is the cell wall, then there is a membrane, and to the membrane here, okay, here this one, this, it is a genomic DNA which is attached to the membrane, and this circular one, these are the plasmids. So we can see that the plasmid DNA, it is in a smaller size than the genomic DNA. Okay, so first step, it is what? The grow the cells and harvest. Okay, and that harvested cell, then it has to be treated with the some of the components. So there is a lysis of the cell. So here there is a use of the enzyme, and that enzyme, it is a lysozyme. So the lysozyme, it is an enzyme which is going to act on the peptidoglycan, and it is going to cleave the peptidoglycan. Then here there is the addition of the EDTA. EDTA, it is a chelating agent, and it is going to do the chelation of the ions, and then there is the addition of the sucrose. Okay, so in this way this solution, it is allowing the isolation of the spheroplast. Now here the spheroplast, it is what there is, okay. Now in this diagram, what you can see, there is an intact membrane, okay, this one, okay, this one it is an intact membrane, but there is a disruption of what, there is a disruption of the cell wall, okay. So the membrane, it has been an intact one, so it is called as the spheroplast. Now this content, okay, this plasmids and this genomic DNA, it has to be come out from the cell. For that purpose, what has to be done, this membrane, it has to be broken down, and to break down this membrane, there is a treatment with the triton X100. Okay, there is a treatment with triton X100. What is this triton X100? It is a detergent. Okay, and we know that the lipids get dissolved into the detergent, and membrane, it is what, membrane, it is a lipid bilayer. Okay, so this lipid bilayer, it can be disrupted, or it can be dissolved, it can be broken when it has been treated with the detergent, and here that detergent, it is a triton X100. Okay, now in this way, now what we have got, we have got the extract. Okay, we have got the cell extract, this one. Okay, there, and when there is a disruption of, okay, when there is a disruption of the, when there is a disruption of the cell wall membrane, okay, when there is a disruption of the membrane, the genomic DNA, it is also getting broken down as it is attached to whom, as it is attached to the membrane. So there will be the fragments of the DNA, they are generated. Okay, this one, genomic DNA fragments, this pink color, it is a genomic DNA fragments, they are generated. Okay, so this, in this cell extract, there will be the presence of the carbohydrates, lipids, proteins, and the broken genomic DNA, and RNA also, it is present. Okay, now here, what is required by us? These plasmids. So here there will be the centrifugation done. Okay, after centrifugation, what is happening? The cell debris, it is going to be in the form of the pellet. Okay, they are going to pellet out. Okay, in that only there will be the large DNA fragments, and in the supernatant there will be the presence of these plasmids. Okay, and now these plasmids, it can be that it can be concentrated with the help of the sodium acetate and the isopropanol. Okay, in this way, okay, in this way, there will be the separation of the plasmid on the basis of its size. That is, the plasmid DNA, it can be separated from the genomic DNA according to the difference in the size. Now, the next method, it is separation on the basis of the conformation. There are the two methods, alkaline lysis method, and second one, it is a CsCl density gradient method. Now, what is this conformation? Here, A, it is a supercoiled structure. B, it is a open circular. Okay, and C, it is a linear one. Now, genomic DNA, it is a circular. The plasmid DNA, it is also the circular. Now, take a rubber. Okay, the rubber, it is a circular one. Okay, and when that rubber, it is going to be twisted. Okay, you are going to twist the rubber on it. Okay, then that structure, it will be called as an supercoiled. Okay, so this, it is a supercoiled structure. Okay, that circular DNA, it is twisted over it. Okay, that it is a supercoiled structure, and this, it is a open circular. Okay, that is, it is a untwisted one. Okay, this is a open circular, and here it is what, there is a presence of this nick. It is a nick DNA. Okay, open circular, open circular, and there is a supercoiled structure or covalently closed structure, supercoiled covalently closed circular. Okay, and here it is a linear, that is the ends of the DNA. Okay, ends of the DNA, they are the free one. So these are the three types of, okay, these are the three types of the conformation. Now, in this, the plasmid DNA, okay, the plasmid DNA, it is present into this supercoiled form. Okay, the plasmid DNA, it is in this supercoiled form, that is the covalently closed circular form. And when there is, okay, when there is a lysis of the cells, at that time, what is happening? the genomic DNA, okay, the genomic DNA, it may become a open circular DNA, or it may get broken down to have the linear fragments. So when there is a lysis of the cells, at that time, the conformation of the genomic DNA, it is going to change. Okay, it is getting a open circular or it is becoming a linear. So now here the conformation of the two types of the DNA, it is going to differ. The plasmid DNA, it is a supercoiled, and the genomic DNA, it may be open circular or it may be the linear one. So this conformation, okay, this conformation, it can be separated out with the help of the method, and that method, it is a alkaline lysis method or it is a CsCl density gradient method. So going towards the first one, it is a alkaline lysis method. So first we have to prepare the cells. Okay, first we have to prepare the cells. That is, we have to grow the cells. And when we are going to grow the cells, while growing the cells, we have to add the antibiotic into the medium. Particularly now this alkaline lysis method, okay, or whatever the methods that we are learning today, that they are for the gram negative bacteria like the E. coli. So here we are growing the E. coli into the growth medium, okay, and when we are growing it, we are adding the antibiotic. Antibiotic, it is nothing but it is a selective pressure. Suppose in the bacterial cell, in the E. coli cell, okay, if there is the presence of, okay, if there is the presence of the PUC18 plasmid, then we are going to add, okay, we are going to add the ampicillin into the medium. So it is going to be a selective pressure. Now, what is this selective pressure? Now whenever, okay, in the home also, or when we are going to do some work, suppose we have a pen, okay, and then if that pen, it is needed to us, at that time we are going to retain it, and if we are not going to need it furthermore, then what we do, we throw it. In the same way, the bacterial cell, it is also going to do the work. Okay, if the plasmid, okay, if the plasmid, it is required by the cell, is required by the bacterial cell, required by the E. coli, then it is going to retain into it. Otherwise, it is going to leave that plasmids. Okay, so here to retain the plasmids into the bacterial cell, there should be the selective pressure, and that pressure in the form of this antibiotic, it is going to be there. Now we are adding the antibiotic into the medium, and the plasmid, plasmid, it is consisting of the antibiotic resistant gene. The bacterial cell, it is going to think that this plasmid, it is required by me because I have to survive into the antibiotic containing medium. So there is a retaining of the plasmids into the bacterial cell. So when we are going to complete our protocol, at that time, the chances of the isolation of the plasmids, they are going to increase. Okay, so here, when we are going to grow the cells, at that time we have to add the antibiotic into the medium, and after growth of the bacterial cells, okay, after growth of the bacterial cells, we have to centrifuge it, and we are going to pellet out the cells. Okay, and that pelleted cells, then we have to dry. It is not a complete dry, we have to just aspirate, we have to just remove the extra medium from the pellet, and we are going to add the various solutions for the lysis of the cells. First one, it is the alkaline lysis solution 1. Okay, this alkaline lysis solution 1, which is called as an TEG, okay, it is consisting of the TsCl, which is a buffer, EDTA, which is a chelating agent. So TsCl, it is going to maintain the pH, and EDTA, which is a chelating agent, it is going to chelate the ions, and the glucose here, it is going to change the osmolarity. Okay, so it is going to weaken the cells when we are going to do the vortexing. Okay, by the vigorous vortexing, okay, there is the instability into the cells, and then we are going to add, okay, then we are going to add the freshly prepared alkaline lysis solution 2, which is consisting of the 0.2 normal NaOH and the 1% SDS. Okay, so this SDS, okay, SDS, it is a detergent. Okay, so when we are adding the detergent, okay, when we are adding the detergent, there will be the complete lysis of the cells. and this SDS, it is going to interact with the proteins and it is going to form the insoluble mass. Now here when we are adding the alkaline Lysis solution 2, that is the NaOH, when we are adding, at that time the pH of the medium, it is going to change from 8 to 12 to 12.5. That is the alkaline conditions they are created. Now when there are these alkaline conditions, during these alkaline conditions there will be the denaturation of the DNA. So when we have added the alkaline Lysis solution 1 and we have done the vortexing, at that time the genomic DNA, it is going to be in the fragments. Okay, it is going to be cleaved into the fragments. The fragments of the genomic DNA, they are formed. That is the linear DNA. Okay, this it is a linear DNA. It is going to be formed when we are going to add the alkaline Lysis solution 1 and doing the vortexing. But here, okay, this blue one, it is what? It is a supercoiled. So there will be no effect over this supercoiled DNA at all. Okay, so here when the pH is going to change at the alkaline side, at that time this linear DNA, it is going to denature. Okay, there will be the denaturation of the genomic DNA, but the effect over the plasmids, which are the covalently closed circular or which are the supercoiled one, the effect over it very small. Okay, and after that we are going to add the third solution. It is the alkaline Lysis solution 3, which is a potassium acetate glacial acetic acid. That is, we are going to again maintain the pH at a 7. So when there is a sudden change into the pH from the 12 to 7, okay, there will be the formation of this tangle mass of the linear DNA. Okay, there will be the renaturation of the DNA. And here the supercoiled DNA, if it has been denatured, it is also getting renatured. Okay, but it is not going to form such a tangled mass. Okay, so here, okay, so here this tangled mass, it is getting pelleted out, and also the impurities like the carbohydrate, lipids and the proteins, it is also going to be settled out when the next step, centrifugation, it is going to be done. Okay, after centrifugation we are getting the pellet, and in that pellet there will be the genomic DNA as well as the impurities from the bacterial cell. E. coli cells, it is going to be at the bottom. Here there may be the presence of the RNA into the solution. Some of the RNA, it may be into this supernatant also. Okay, so in this way, okay, in this way, there will be what? Plasmids, supercoiled plasmids into the supernatant. Now this supernatant, it has to be collected, and after collection there will be the plasmid recovery, and the plasmid recovery, it is done by adding the 2 volume of the ethanol. Okay, so ethanol, it is a dehydrating agent. Okay, so it is going to dehydrate the plasmid DNA, and the plasmid DNA, now it is going to pellet out, or um, it is getting precipitated, and then after centrifugation we are getting the pellet of the plasmid DNA, and which is going to be washed with the 70% ethanol, and the pellet that we have got, that pellet, then it is going to be dissolved into the TE buffer. Here in the TE buffer we have to add the RNase A, because a small amount of the RNA, it may be the impurity which is present into the plasmid DNA suspension. So that RNA, it has to be cleared, and for that purpose there is the use of this RNase A. Okay, this RNase A, it can be added when we are doing the lysis of the bacterial cells. So there will be the complete removal of the RNA. Okay, so here, okay, in this way there will be the isolation of the plasmid by the alkaline Lysis method. Okay, here for the for a highly pure form of the plasmids, okay, the supernatant that we have got, it can be treated with the phenol chloroform, and we can get the highly pure plasmids after the alkaline Lysis method. The phenol chloroform, it is an optional option if for the highly pure plasmids by the alkaline Lysis method. The second method, here it is a CsCl density gradient method. Okay, CsCl density gradient centrifugation. It is a centrifugation, calcium chloride density gradient centrifugation, and here the gradient of the CsCl, that is the calcium chloride, it has been maintained into the centrifuge tube. Okay, like here 1.55 to up to the 1.75. In this way there is a creation of the gradient, and the cell lysate that we have got, that cell lysate now it is suspended into the CsCl gradient and allow it to centrifuge. And after centrifugation you are getting the three bands. The topmost, it is a protein. Okay, here there is a floating of the proteins because its band density it is a least one, and here we are getting the pellet of the RNA, and here at the 1.70 we are getting the band of the DNA. The bond density of the DNA, it is the 1.70 grams per centimeter cube. So here we are getting the band of the DNA, and this DNA it can be removed with the help of the syringe. Okay, but as it is a DNA, it is consisting of the plasmid DNA as well as the genomic DNA. So how to separate out this plasmid DNA and the genomic DNA by the CsCl density gradient method? Here, before going to the CsCl density gradient, the cell lysate, it is going to be treated with the EtBr, and now the method it is the EtBr-CsCl density gradient method. Okay, now we know that the EtBr, it is an intercalating agent. Okay, EtBr, it is a intercalating agent. So this EtBr, it is going to intercalate in between the bases. Now, when we are treating the EtBr with the DNA, the highest amount of the EtBr which is going to bound with the DNA, that DNA, it is a linear DNA. Okay, the EtBr, it is getting intercalated at a high concentration in the linear DNA, then it is going to bound with the open circular DNA. Okay, and the least it is getting bound with the supercoiled because there is a less spaces to get bound with the plasmids. Okay, so the plasmids, they are in a supercoiled form, so the least amount of the EtBr, it is getting bound with the supercoiled plasmid DNA. So what this EtBr it is going to do, it is going to reduce the bond density. Okay, so bond density of the linear or the open circular DNA, it is going to be reduced by the 0.125 grams per centimeter cube, and for the supercoiled it is of about the 0.085 grams per centimeter cube. So here the bond density of the two types of the DNA, now it is getting changed, and as it is getting changed, it is going to form the bands at the two different places. Okay, so here the high decrease into the band density, so the band it is going to move upward side, and the supercoiled DNA, it has a least change into the band density. Okay, so it is getting separated from the genomic DNA, and now here this band of the plasmid, that is the supercoiled DNA, it can be removed with the help of the syringe. Okay, now into this plasmid DNA. Okay, into this plasmid DNA, there is a binding of EtBr. So that EtBr, it has to be removed, and to remove that EtBr, it is treated with the organic solvent like the N-butanol. Okay, so when we are going to shake the butanol with this plasmid DNA suspension, then we are getting the plasmids into the aqueous solution. Okay, aqueous phase, and the EtBr into the organic phase. In this way, we are going to separate out the EtBr from the plasmids. Still, into this aqueous phase with the DNA, there is a presence of the CsCl. Okay, there is a presence of the CsCl. So this, okay, this aqueous solution, now it is going to be in the dialysis bag, and then the dialysis it is going to be done so that the CsCl it is going to diffuse out from this aqueous phase, and we are getting the pure plasmids into the dialysis bag, and now we are going to remove the dialysis bag, remove the solution, and in this way we are getting the highly pure form of the plasmids. Okay, what we are getting? We are getting highly pure form of the plasmids. Okay, so EtBr-CsCl density gradient centrifugation method, it is the choice of the method for the highly pure form of the plasmids. Okay, these are the two methods according to the conformation. Yes. Now, next it is a boiling method. Okay. First, we have to prepare the cells. Okay. And for the preparation of the cells, for growing of the cells, what we are going to add into the medium? Yes, we are going to add the antibiotic. That is a selective pressure we have to maintain. Then we have to collect the pellet of the cells by the centrifugation. And here, now here, the method, it is what method? It is a boiling method. So here we are going to add the solution, and that solution, it is a STET solution. This solution, it is consisting of the sodium chloride, TrCl, EDTA, Triton X100 or the Twin 20. Sodium chloride, it is a salt, TrCl, it is a buffer, EDTA, chelating agent, Triton X100. What it is? It is a detergent. Twin 20, it is a detergent. Either Triton X100 or the Twin 20 we have to use, and then there is a vortexing. Okay. After vortexing, there is a weakening of the cells. Then this mixture, it is treated with the lysozyme. Okay, so lysozyme, what is it? It is the enzyme. Okay, on what it is going to act? On the peptidoglycan. That is, it is going to act on the cell wall, and it is going to cleave the cell wall. And then we have to keep this suspension into the boiling water bath, and so that there will be the lysis of the cells. Then we are going to cool it, centrifuge it, and we are going to collect the supernatant. And in the supernatant, now there will be what? There will be the presence of the plasmids, and that plasmids, it can be recovered by adding the sodium acetate and isopropanol. So here, when we are adding the sodium acetate and isopropanol, actually we are going to dehydrate the plasmids. So plasmids, they are getting precipitated, and after centrifugation we are getting the pellet of the plasmids, which is going to be washed with the 70% ethanol, and then it is going to be dissolved into the tea buffer after drying, and there will be the addition of the RNase A. RNase A, it is the enzyme which is going to cleave the RNA. So the impurities of the RNA which is present with the plasmids, they are going to be removed. Okay. In this way, there are the various methods for the isolation of the plasmids. Okay. So we have discussed about the isolation of the plasmids uh, on the basis of the size according to the conformation. In the conformation, we have seen alkaline lysis method and the CsCl density gradient method, and here very different method, that it is the boiling method. It is used very frequently. There are the various methods. There is the chromatography also, which is used for the separation of the plasmid DNA from the genomic DNA. Okay, I think you may have cleared the idea about the isolation of the plasmids. If you like my video, please subscribe my channel and also share my videos with your friends, so the knowledge, it is going to spread. Thank you.